Hi, I'm Alison Irwin, a student at the Natural History Museum. My PhD project focuses on the evolution of eyes within Stromboidea. This is a marine group of snails, and what makes this superfamily particularly interesting is its wide range of visual structures and capabilities that are found within the group. The diversity of eyes in Stromboidea can be seen from the outset. Within this superfamily, 3 out of 5 families possess large and colourful camera-type eyes at the end of long mobile eye stalks. This includes the family Strombidae, pictured here. Earlier studies have shown that the optics of the eyes of these 3 families are very similar in sophistication to that of squid and carnivorous snails called heteropods, which both rely heavily on vision for prey capture. With these comparisons in mind, it's incredibly surprising to find similarly well-developed eyes in slow-moving herbivores like the strombids. By comparison, the eyes within the remaining 2 stromboid groups, Aporrhaidae and Struthiolariidae, are small and darkly pigmented on very short eye stalks near the base of cephalic tentacles. These eyes presumably provide much poorer vision than the other stromboids, which you can't really see anyway, so I'm just going to zoom in. While most of the large-eyed families can be found in tropical shallow waters, the smaller-eyed families tend to be found in deeper waters or in much murkier waters than the tropics, such as our own lovely UK. This contrast in eye types, as well as the extremely good eyesight of some families, is why Stromboidea is an excellent group for eye evolution studies. However, little morphology or molecular data currently exist for almost all of these families, and there is no existing phylogeny that includes all groups within Stromboidea. In order to investigate eye evolution in this group, we need to collect eye morphology data using different techniques, such as histology. We also need to build a robust phylogeny of the superfamily using mitogenome and Sanger sequencing data, and use this phylogeny as a framework to investigate trends in eye evolution, including when large eyes evolved. This is what I'll be presenting today. So first, studies into the varied eye structure and functions within Stromboidea. How can we compare visual capabilities between very different groups? First, we need to understand what animals can see, which we can estimate from morphology data. Understanding how an organism perceives visual scenes requires knowledge of that animal's colour sensitivity. Also, temporal resolution, which is whether you can track rapid changes in a scene, or if you just see casting roadrunner blurs. Thirdly, spatial resolution, the ability to perceive detail, and finally, sensitivity, the ability to distinguish contrast. In this talk, I'm going to focus on spatial resolution to compare visual acuity between families. Spatial resolution can be measured from histological sections, represented as the interreceptor angle. This angle is calculated using the focal length and the separation of the photoreceptor centres. What this diagram shows is that the animal will be able to distinguish the alternating black and white bars if the angle from one photoreceptor falls on a black bar, and the angle from the next photoreceptor falls on a white bar, as shown on the left. If this is not the case, as shown on the right, the bar will appear to be a mid-grey blur, like so. So, an animal with a large interreceptor angle will see a blurred image as opposed to one with a high level of detail. Histology sections here show a spherical lens typical of marine animals with camera-type eyes, with a wide aperture to allow plenty of light to enter for image formation. The blown-up image on the right shows the retina with photoreceptor cells packed tightly together, containing a layer of photopigment cells. The interreceptor angle was calculated from the separation of the photoreceptor centres and the focal length using these images. The large eyes from the family Strombidae are here compared with the smaller eyes from Aporrhaidae. In Aporrhaidae, the retina contains far fewer photoreceptor cells, meaning the image formed will be blurrier, like if the computer screen you're watching this on had fewer pixels. I found the interoceptor angle to be 2.5 degrees in Aporrhaidae and 0.5 degrees in strombid species. So, strombids theoretically have a resolution 5 times finer than that of Aporrhaids. We can compare these values of the interoceptor angle to the resolution in other animals. The smaller the angle, the higher the resolution, with humans placed at the top of this table. In mollusca, the octopus eye has the highest resolution, which enables sophisticated behavioural tasks, including communication and fast prey pursuit. From this, we can see that strombids have a spatial resolution of twice that of a worker bee and 9 times that of the periwinkle Littorina. But how can we use these values to understand what these animals see in their environment? It can be difficult to visualise, but using these interoceptor angles, we can blur this photo of a reef to indicate what a strombid and other taxa can see in terms of resolution. From this, we can clearly see that strombids are able to resolve small objects in their environment, and more so than you would expect for a herbivorous marine snail compared to these two predators outlined in red, and especially considering both Littorina and Aporrhaids are also herbivorous marine snails. These differences in visual capabilities and structures between large and small-eyed families in Stromboidea highlights the need to look into this from an evolutionary perspective. This brings us to the molecular phylogenetics section of this talk. To find the transition point of large-eye evolution, we first needed to build a phylogenetic framework. We used complete mitogenome data to robustly test the relationships between representatives of all 5 families in Stromboidea, as well as from another family, Xenophoridae, which also has small eyes. We used Illumina whole genome sequencing to generate reads, which were first assembled to reference mitogenome sequence, and that the largest area of high coverage of the contig was extracted and assembled iteratively to the reads. The consensus sequence was then annotated using mitoZ and circularized. In our analyses, we included mitogenome data from other senior gastropod groups to test the monophyly of Stromboidea. Results showed that the monophyly of Stromboidea is only recovered if Xenophoridae is included in the superfamily, something which so far has been viewed as controversial in taxonomic studies. So, zooming in on Stromboidea, this analysis was constructed in Mr. Bayes using all mitochondrial genes, including ribosomal genes, and 2 nuclear genes, 28S and 18S. This tree shows 2 clades, one of small-eyed families and one of large-eyed families, indicating that large eyes evolved somewhere here along this branch. Sign of sequencing of 4 genes was used to generate this tree in Mr. Bayes, which includes representatives from several key taxa in Stromboidea. The eventual objective of this work is to obtain sequence data from as many stromboid species as possible, and then map on morphological data. These initial results demonstrate how we can begin to investigate trends in eye evolution across the tree. For example, the measurements I've shown here are absolute eye size relative to body size proxy, and whether species have been observed to be active during the day only or during both night and day. As you can see, this is a work in progress, but these initial results show large clusters of species that have been observed to be active both at night and during the day, with large eyes relative to body sizes, as I've outlined in black. Large eye sizes increase the amount of light collected and absorbed, which increases the sensitivity of the eye. These traits may therefore be adaptations to activity in dim light environments. By plotting this data, we can begin to understand more about the trade-off between visual sensitivity and resolution, and how these eyes have adapted. Other morphological data, as well as habitat and depth data, can also be plotted on trees to investigate trends in eye evolution. Through this talk, I've shown that anatomical studies provide valuable insights into how strombids view their surroundings. Histological studies predicted that eyes would resolve a shape of about 0.5 degrees. This shows that strombids have a high spatial resolution and can resolve much smaller objects in their environment compared to their smaller-eyed relatives within Stromboidea. Phylogenetic analyses using mitochondrial genome data resolve two clades, a small-eyed clade and a large-eyed clade, meaning that we can target where large and complex eyes evolved in the group. Xenophoridae was resolved as sister to the small-eyed families in Stromboidea, and therefore belongs in this group. Finally, plotting morphological data onto trees can help us to investigate eye evolution, and we can better understand the diverse structures seen in the superfamily. To further develop this work, I will use histology and serial block-face SEM data to do 3D reconstructions, which will help us to understand the diverse eye structures across the superfamily. I will also increase the database of morphology measurements to plot on trees and the number of Sanger sequences I have available. Our final aim is to time calibrate trees in order to estimate when adaptations occurred. Therefore, I am also going to collect fossil first and last appearance data for key taxa. This will help to resolve key transition points in the evolution of these amazing eyes. Lastly, I would like to thank my supervisors, all those who have been involved with facilitating molecular lab work, the Smithsonian for hosting me for histology work, and all those who have loaned me snails. And you guys, thank you very much for listening.